Thank you. Thank you for having me. And I look forward to sharing with you today the information that I've learned over the last 25 years in terms of neuroscience and brain plasticity. So today I'm going to be talking about, the summary is that I'm going to be talking a little bit about sugar and the controversy about whether it's addictive or not. I'm going to share with you all of the research from my lab that looks into the neurobiology of addiction. And really, sugar, is it really anything like alcohol and nicotine? I have to say this is exactly how I felt when I was doing my alcohol research. I'm going to tell you a little bit about the neuroscience of sugar addiction. And then I'm going to switch to another mode where I'm not just going to talk about the reward substance sugar. I'm going to talk a little bit about the reasons for addiction in the first place and how stress and adverse childhood experiences lead people to eat sugar, drink alcohol, etc. And I like to call this the brain scales of justice. On one side we have stress, on the other side we have reward, and the brain's always balancing these two, and I'm going to talk a little bit about that. Then I'm going to finish the webinar telling you about the latest cutting-edge research where we're really able to now look inside the brain, and we've been able to dissect it much more than we've ever been able to in the past. I'm going to tell you about the role of medications, genetics, epigenetics, and how we can really embrace brain plasticity, or what we like to call neuroplasticity, to reduce addiction. The bottom line is we are all getting fatter. There are some estimates that by 2025, almost, if we don't change current ways of eating and sedentary behavior, etc., 80% of people are going to be overweight or obese. Now, there is lots of causes for this, and we do know that one of them is the increase in intake of fructose that's contained in soda, sweets, sugar, and of course alcohol. And everyone has a different argument about this, and today I just want to talk about one of these components, and that's fructose. So sugar, or sucrose, is made up of fructose and glucose, and fructose has really entered and become embedded in our food chain over the last 40 or 50 years, starting in the 1970s. We all know that people struggle to lose weight over the long term, and the cost of this increase in being overweight and obese is enormous and has been shown to play a significant role in cancer, diabetes, dementias. It decreases our overall well-being and productivity and causes mental health problems such as depression. But the thing that really occurred to me in all of the conversations around eating and drinking and obesity is that the role of the brain and neuroscience seemed to be relatively left out of these equations. Everything else was considered, but the role of the brain. We all know that this is a struggle for some people, not for everyone, but we do know that diets fail. There's tons of research evidence to show that diets work in the short term, but over the long term, most people struggle. And this is just a pictorial demonstration of the struggle that people face in this regard. I became a neuroscientist because my sister suffered from a mental health problem in 1989. And she was in a lockup ward, and I didn't like how she was treated. So I went back and became a neuroscientist. I was a pharmacist at the time. And I now put on my pharmacist hat as I entered into the neuroscience field. But in the last 5 years, I've really changed my research direction from developing medications to now trying to understand how we can enhance the ability of the brain to change, brain plasticity, that is. So I spent a long time leading a lab at the Gallo Research Center at the University of California, San Francisco, and then I was recruited to the Translational Research Institute at IBI, which is where my research lab currently is. Now the bottom line is, and this is my hypothesis and the argument I'd like to make with you today, and you can weigh up all the evidence yourself. But from the last 5 or 7 years of research in my own laboratory, I've come to the conclusion that sugar is addictive. And just like all the other addictions that I've studied over the last 20 years, it really ends up being a brain disorder. And there are some elements of it that can be treated with medicines, but not all of it, but just some of it. This is research here I'm showing you from Neurobiology's lab at the National Institutes of Health. And what we're seeing here is brain images of people that have cocaine addiction, alcoholism, and addiction to nicotine or tobacco lined up with people that are overweight or obese. And this is a BMI or body mass index over 30. Now, what you're looking at in this picture in yellow and red is dopamine receptor availability. And you can see that it's changed from normal down the bottom panel compared to people with addiction. What's the bottom line? What does that mean? It means that to get the same level of reward, you have to keep taking more and more of the same substance. My lab so I've been studying alcohol and its impact on brain structure and function for a good 20 years. And the really amazing thing and totally shocking thing to us, we were using sugar as a control for all of our experiments, just so we could replicate the behavioral effects of alcohol. And the bottom line is we started to see that long-term sugar consumption, at least in animal studies, changes the structure and function of the brain and activates specific receptors that both alcohol and nicotine activate. And I'm going to show you that evidence as we move through this presentation. Now, I think we all know that there's a strong neurobiology that underlies addictive behaviours. And I am sure you've learned all about these different brain pathways and circuits before. And I'm only going to focus on a couple of them today. And that's the amygdala, the ventral tegmental area, and nucleus accumbens. The amygdala kind of processes fear and stress and other aspects of negative behaviours. The reward process or motivation is handled by the accumbens and the VTA, and these are sometimes referred to as the pleasure and motivation pathways. All of these signal to the front part of the brain, the prefrontal cortex. That's where we make our short-term decisions and set our long-term goals. So if you're noticing with any of your patients or clients that they have a hard time implementing all of the strategies that you're telling them to implement, and inside your head, you're thinking, why can't they just go about and do this? Well, I hope by the end of my presentation today, you'll see that they don't actually have the neural networks or the circuitry inside the prefrontal cortex, the top part of the brain, to be able to execute on all of the plans you're telling them to make. I think there's nothing I have to do to say to you that alcohol and nicotine is addictive. We all know it is. And this has been done for years and years and years. Now, the thing that when I started my lab that was very clear to me was that many alcohol-dependent subjects smoke. But if you smoke, you're not necessarily addicted to alcohol. So this is an interesting observation. So there's so much genetic, behavioral, and pharmacological evidence that demonstrates there are common genes associated with susceptibility to develop both alcohol and nicotine dependence. These are the papers here that I've listed for you. We know in animal and human experimentation that chronic nicotine administration increases ethanol consumption. We know that it can, if you give nicotine to abstinent people from alcohol, you can reinstate alcohol-seeking behavior in animals and humans. And lastly, a very compelling piece of evidence is if you use a non-selective nicotine receptor antagonist, you can actually decrease ethanol and nicotine intake in animals and in humans. So what's the bottom line here? Well, what's the common molecular pathway that underlies the comorbidity of alcohol and nicotine dependence is the neuronal nicotinic receptor. It's really a common pathway. This receptor is a ligand-gated ion channel made up of multiple subunits. It's the receptor that drugs such as Varenicline or Chantix, marketed here in the US, bind to. These are pentameric ion channels, and both we and others have shown that nicotine and acetylcholine, which is the endogenous ligand that binds to nicotinic receptors, activate these channels. inside the brain. These receptors are actually located in all the brain regions associated with addiction. And furthermore, both our ethanol, sugar, and nicotine can activate this limbic brain system via these receptors to affect dopamine transmission, glutamate transmission, and GABA transmission. So in my lab, we've been studying ethanol consumption for some time. The reason I'm telling you about this is because it is to give you the underlying reason of how we got to sugar and why we think it's addictive. These are the type of experiments that we do in the lab. Now, how our experiments differ from others is that we actually study animals consuming ethanol or sucrose for very long periods of time because we're trying to mimic the human situation. We were very fortunate to be able to get animals to drink large amounts of alcohol by changing the protocol for inducing ethanol addiction inside the lab. This is where animals have a choice between drinking water or ethanol over a 3-hour period, and they get this every second day rather than every day. So that day off allows us to drive an increase in ethanol consumption. We call this the drinking in the dark model. In this next slide, what I'm going to show you is what happens if we just give the animals 10% ethanol every day. You can see there's very little escalation in ethanol consumption. However, as soon as we move to an intermittent access, day on, day off, we see a remarkable escalation. And I think that you'll hear this from people that drink alcohol also. That withdrawal drives the craving, which drives us to take more alcohol so we can reduce the effects of withdrawal. So we've developed a very nice animal model in the lab where we can actually study and try and understand and dissect the molecular pathways that are underlying this escalation in drinking. So in 2007, I got very lucky, and I was working with in a collaboration with Pfizer, and the drug hadn't been marketed yet, but my lab was the first to get Varenicline or Chantix into a lab. And this drug is not metabolized by the liver, but it competes with nicotine and acetylcholine at nicotinic receptors, and this leads to a decrease in dopamine and the accumbens. We believe this is how it works to quell some of the cravings associated with nicotine addiction. At this time, 10 years ago, no one had looked at alcohol consumption. And my lab had set up these really amazing animal models, and we were the first to be able to demonstrate that Varenicline could actually reduce ethanol consumption in animals, but not if the animals were social drinkers, only if the animals had been drinking ethanol for a long period of time. The beauty of these experiments was we were able to then move it into human studies pretty rapidly because the drug had been approved by the FDA. So we went on to then show that Varenicline could also reduce alcohol's activation of the limbic system, or the VTA, in humans. So these were very small proof of concept clinical trial type studies. Then we went on to conduct a number of clinical trials looking at the role of Chantix in alcohol consumption in humans. And these are small studies, but we've been able to replicate them in multiple labs across the world. And that is that Varenicline indeed can reduce alcohol consumption in humans also. So this is not what the presentation today is about. But while we were doing all these studies, as I alluded to, we had sugar set up side by side next to the animals drinking alcohol. And I don't know how you feel about this, but I was very much of the mindset that there's no way sugar is addictive. And that's the nature of science and serendipity, is that we then went on to study it because we got these really interesting findings. So just like I alluded to before, we set up these experiments just like replicating exactly how we do the experiments for ethanol consumption. And then really surprisingly, we discovered that just like for alcohol, Varenicline or Chantix could reduce long-term sugar consumption, not short-term. See here in 4 weeks of drinking. Over here, you'll see 12 weeks of drinking, and now the drug becomes more effective. That was really, really surprising to us. In fact, it was so surprising we didn't believe the original set of data, and we went back to replicate it over a number of years. And that turned out to be the PhD thesis of one of my students. We went on to then look at not just Chantix, but also a number of nicotinic drugs such as cytosine, which is marketed in Eastern Europe for nicotine cessation, and saw very similar findings. And this led us to think and ask many questions, which we're still now doing. This is the majority of the work in my lab right now. It's focused on this question: how can sugar be activating nicotinic receptors in the brain? We know nicotine is one of the most addictive drugs in the world. And it's very straightforward to understand nicotine binding to nicotinic receptors and activating addictive pathways. But sugar, really? How is that possible? Our current hypothesis is that sugar, and we're not talking just any amount of sugar, we're talking overconsumption of sugar, a lot of sugar, the sort of sugar that changes the body and causes metabolic syndrome, for example. So I'm just trying to go back to that slide. Now, as I alluded to earlier, nicotinic receptors are pentameric ion-gated channels made up of different types of alpha and beta subunits. And as I said earlier, we all have an endogenous ligand, which is acetylcholine. Our current hypothesis is that sugar increases the levels of acetylcholine inside the brain, and this activates nicotinic receptors, just as nicotine would. So in collaboration with Dr. Quick at Stanford Research International, we went on to ask questions that are very similar to what we've asked to do with alcohol. And lo and behold, we found, and this is really the first discovery we made, was that after animals are drinking sugar for a long time, there is a change in the expression of nicotinic receptors in the brain, such that you see an increase in alpha-4 beta-2 nicotinic receptors in the nucleus accumbens, for example. All of these studies have been published just in the last 2 years. So if you're interested, on each slide, I've put a reference to where you can find more detail about each of these studies. Also, my lab focuses very much on looking at neuroplasticity or brain plasticity. There's things that cause both the brain to have positive neuroplasticity, that is, to be improved by good things, which some people might refer to as brain resilience, or negative neuroplasticity, things that make the brain more fixed or not as efficient, such as looking at the effects of ethanol and now sugar on the structural and functional properties of the individual neurons after consuming these substances for a long period of time. So in previous studies that followed on from our alcohol studies, we were able to show that ethanol changes not just the structure of the brain, but also the functional properties, specifically of the two important brain areas involved in the neurobiology of addiction, such as the prefrontal cortex and the amygdala. And I'm not going to go through these in detail because we don't have enough time, and all of the papers are available here. I've referenced each of these pieces of work that we've done and we continue to do. But this picture is just to show you how ethanol, and this blew my mind, to be really honest. Look at this. You see water at the top, animals consuming water, versus animals consuming ethanol over a long period of time. We extract the brain, we slice it, and then we label it with neurobiotin, and then we do both measurements of how the dendrites have changed and how many synapses have changed, and we get right down to whether it's in the top part of the neuron or the bottom part of the neuron. And I don't know how hard it is for you to see this on your screens, but if you look at the bottom panel here with ethanol, you can see a remarkable change in the structure of the neuron in the prefrontal cortex. I don't know how you feel about this, but knowing this from my point of view changes everything for me in terms of how I run my research lab now. You can see down the bottom is we've measured each of those different contact points. We've measured the different lengths. It's just very laborious but very important work. And this was published in 2016. The key here is not just the structure changing. This structural change actually leads to a functional change. in the way those neurons are firing. And you can see that in these images on this slide here. You can see animals consuming water or consuming ethanol. You can see changes in the properties of how those neurons fire. So this is why this is such important work. We were combining not just imaging and structural changes, but we were combining it with functional changes. So these structural changes are leading to functional changes. Okay, so what about sugar then, and sugar's effects on the brain? So it was not a very big step for us to then move to these experiments to focus on sugar or sucrose. And in the first set of experiments that I'm going to show you here, we're actually looking at changes in the structural properties of the amygdala, the area that we all know processes fear and stress and other things. In these images here at the top panel, you can see 4 weeks of sucrose consumption. The dots in the middle represent the basolateral amygdala. And what we're looking at here is the parts that we went on to study. The dots represent the neurons that we studied. And if you can see down the bottom, you see 12 weeks of consumption, and you can see the significant changes, either an increase or a change in location or a decrease in the types of structural changes we're seeing in the basolateral amygdala. This is published. This is very recent work from our lab, and we continue to do this because we're really shocked to find these discoveries. That's the amygdala. We went on to look at changes in the nucleus accumbens, and as you can see, the changes aren't as dramatic as in the amygdala, but certainly we're seeing changes in the structural property of the nucleus accumbens. These are in the dendritic medium spiny neurons of the accumbens. And here is just some more data to show you just how significant these changes are, where we see a decrease in dendritic length in the distal branch of these neurons from the BLA. I just want to make a point here. These are in long-term sucrose-consuming rats compared to control rats. These are not human studies, these are animal studies, because it's very difficult. I don't think anyone will allow us to do that. But I just want to make the point here before I move on. This is remarkable to me. This was completely unexpected finding. This is not what I was looking for in our lab. These were controlled experiments. But obviously, I think it doesn't take too much to make the next step in how come we haven't studied more about the role of sugar and addiction in the development of obesity. It's just one component of a very complex problem, but this problem is not going away. There's just been a steady increase in the percentage of people either overweight or obese. And there's lots of reasons and many causes for that, including genetic susceptibility, like how things were inherited, how we inherited different susceptibilities from multiple generations, to the type of food and the amount of food that we eat now, whether it's high-fat food or sugar-sweetened beverages, but both the size and quantity. We're much more sedentary now, and we sleep, often sleep-deprived, and adverse childhood experiences happening during childhood. Each of these underlie the development of obesity. But I've hoped, in summary, I hope that you can see that sugar is one component that we are over-consuming, that has become embedded in the food chain. 75% of all food now that is processed in the US since 1968 has become embedded with fructose, or high fructose corn syrup. We now know that that activates the same brain circuits as alcohol and nicotine, and it's making the same structural changes as alcohol and nicotine. And we haven't proven yet, but we have a hypothesis that it's increasing our endogenous ligand acetylcholine in the brain to activate these receptors and change their expression and their localization. We also know from other studies in other labs that fructose component of sucrose or sugar activates the hypothalamus and makes people feel hungry or increases appetite. So you can see there's a double whammy. Sugar is not just calories, it also is causing things that change the structure and function of the brain in a similar way to alcohol and nicotine, which makes it incredibly difficult to give it up. And many people don't believe this, and I've been approached and emailed by many people that really do identify with being addicted to sugar. They're not addicted to alcohol, but they feel addicted to sugar, where they get withdrawal. And when they approach their healthcare professionals, they're told that it's not addictive. So sugar can change the brain. So the parts of the brain that are changing, interestingly, in the prefrontal cortex and in the amygdala, suggests that it's affecting impulse control. Just as you see with alcohol addiction, there's tons of studies demonstrating that alcohol affects the prefrontal cortex, which affects impulse control, which makes it harder for people to delay gratification. That is, take a short-term reward over a long-term reward. We're seeing the same for sugar. Furthermore, sugar is activating the part of the amygdala that's making the brain more reactive to stress. So even though it might reduce stress in the short term, it ends up increasing stress reactivity part of the brain, just like alcohol does over long term. And lastly, and double whammy, sugar makes us hungrier, so therefore it makes us want to eat more food at the next meal, whether it's a high-fat food or others. So this is all really interesting, and as usual, we're focusing on the reward part of addiction. That is the things that people are doing to make themselves feel better. But what are really the underlying reasons for addiction? What is it that drives people to drink in the first place, or overeat sugar in the case of obesity and other things? What makes people drive for addictive substances? I'm going to turn my hand a little bit to that right now during this presentation. And because of all of my findings, and because I've studied the brain for 25 years and discovered that the brain has a massive capacity for change using neuroplasticity, but it's locked down by stress and sugar's effects on the brain and the body, I decided that people didn't seem to have very much information about how the brain worked in their hands. And that's why I've written these books for a lay audience that discusses each of these things I'm talking to you about today. One about the effects of stress and sugar on the brain, 2 about what to do about it using neuroplasticity, and 3, all about all of the things that happen from adverse childhood experiences that end up wiring the brain for addiction in the first place. And if you're interested, these books are available at my website. You can learn more about my podcast there, and they're all available on Amazon. I just released Smashing Mindset and Trace It to Erase It in 2017, and my first book, MiGGI, which is all about the amygdala and addiction, I released in 2016. Smashing Mindset is just an updated version of MiGGI Matters. So now let's turn our focus on the underlying causes. I think this is really important. We've spent, and I'm guilty of spending a good 22 years studying the reward, pleasure, and negative side effects of the drugs that people are consuming for addiction. However, the brain is an amazing machine and is always putting things in balance between stress and reward. A foundational study by Coletti and Ander that was started in 1985. It wasn't called the Adverse Childhood Experiences Study back then. But what they did was they're interested in trying to help people reduce obesity, because obesity was on the rise in the 80s and it was affecting, their health system. What they discovered was during this study was as people started to lose weight that were obese, they started to drop out of the study. So they went and interviewed these people, and they discovered that they mentioned that as they started to lose a lot of weight and were becoming very successful, they all of a sudden had to deal with the trauma and the reason they were eating in the first place. So they reframed the study, the Adverse Childhood Experiences Study. I know you've probably heard a lot about this study because there's Nadine Burke is doing an excellent job of getting this integrated into practice. And for me, it seems so important, anyone that's dealing with addiction or other things, people should know the ACE score of their clients and patients. Furthermore, we have a genetics. We don't start from a blank slate. We have a brain that's been wired over millions of years, and that brain has been growing up in an environment and is being wired by experience. So every brain is completely different. So one way I like to help you think about this and why it's so important is, do you know what's happening inside your brain? Like people listening to this webinar, can you think what is happening inside your brain when you're stressed out? what are the things that you do to make yourself feel better in terms of food, sugar, and alcohol at the end of the night? Is it to go to the fridge to have a glass of wine? Is it to reach for that chocolate bar or the candy? This is very important to understand for yourself first so that you can get into the minds of the people that you're looking after. Stress is so important to manage first to reduce the impact of addiction. Stress reduces brain activity in the brain regions that we know drive reward seeking. What happens as stress changes brain circuits? It makes people not being able to think clearly, plan clearly, or have control over their impulses. We know that stress predisposes the brain to depression, anxiety, and addiction. So when we're talking about sugar and alcohol, they're the outcome from the way the brain had been wired from adverse childhood experiences. I'm not going to talk about this for very long, but just to let you know that this is an amazing study, and I really encourage you to look at it. You can actually also get your own ACE score. Why am I laboring this point? Well, because adverse childhood experiences, this is in graduated college students some years later, disrupt neural development. This leads to the adoption of health risk behaviours such as alcohol and addiction to sugar. These are the type of questions that, these are sort of stresses that were measured. It's a really easy survey to do. These are some of the questions that are asked, and you can go to this website, acestudy.org. It takes very little time to do it, but it gives you a great insight in why it's so difficult for some people to manage stress compared to other people because of the way their brains have been wired so differently. I find this a shocking study, but as a neuroscientist, it's really illuminating to me because it kind of partly answers the question, why are we also addicted to something? And that's because even in people that have had the best start in life, 62% of adults have experienced at least one ACE, and 1 in 6 people have experienced up to 16% of ACE. And this is just not statistics here. You know, the amazing thing is they went on to demonstrate that depending on the amount of ACE you've experienced, it's really highly correlated with whether you'll become an alcoholic later in life, whether you'll go on to develop obesity, this is a BMI above 34. These are percentage changes as your ACE score. And I just want to point out, these are high-ish ACE scores, but people that you're dealing with often will be up in the 10 to 17 range because those people have been excluded from this study. I hope that is alarming enough to make you realize why each person you're dealing with is so different. That's because they have a different genetic start. And the way that adverse childhood experience, or stress as we call it, has wired the emotional part of their brain is completely different. And this part of the brain is activated in milliseconds before we're even aware to generate the freeze, flight, and fright response to physical threats. That's why people that use drugs or sugar or alcohol to reduce the impact of all of the things that they're seeing as physical threats is constantly, repeatedly stressing their brain. So the bottom line here is, and because it's so important, and so the brain is such an amazing machine that can be shaped by the current experience and can be reshaped by a new experience. What I did was to write a book that the average person can understand, trying to democratize this information. MiGGI, I called it MiGGI, this emotional part of the brain, to help people realize that it is the way the brain's being wired by these experiences. And just as it's been shaped by previous experiences, it can be reshaped using neuroplasticity for future experiences. I think this is so incredibly important to help us improve our way we're treating addiction currently. I think it's really clear from seeing all the structural and functional changes in the brain that just using a drug is going alone may have some helpful parts for certain parts of addiction, but in general, a drug is not going to be able to, one drug or a combination of certain drugs isn't going to be the be-all and end-all to help people overcome the impact of adverse childhood experiences and how it's impacted the brain that's led to addiction. As I like to call it, the emotional part of the brain is always balancing stress and reward. Because stress or adverse childhood experiences have affected brain development or can lead to negative consequences to the brain itself, the brain doesn't like that, so it's always seeking to balance that with reward. So if I use me as an example, or people that have had minimal adverse childhood experiences, we might replace that with something like work, career, money, retirement, divorce, for example, as things that will activate that part of the brain. And then to mitigate the effects that has on our brain, we will then be seeking things like wine or beer or alcohol at the end of the day, or high-fat food or sugar or chips. Everyone has their thing, as I like to say. This is why stress imprints the body. So if we're not handling stress in the way it's wired the brain using neuroplasticity principles and reducing sugar intake or alcohol intake, this ends up imprinting all over the body. And that's been well known. But the key here, the clinicians and other people helping people, and why it's so difficult to apply one principle to one person, every brain is different. Genetics, environment, and experience. We know that genetics for alcohol addiction can explain something like 60% of alcohol. We know that if you grow up in an alcoholic environment, you're much more likely to become addicted to alcohol. If you're fed more sugar during childhood, the chances of getting metabolic syndrome or addiction to sugar are much greater. So these things are always constantly rewiring the brain. I like to say that there's an epigenetic period that happens between the ages of 0 and 3 and 10 and 14. So why adverse childhood experiences lead to addiction is because there's a period of time between when the brain first starts developing until the age of 3, where the brain is neuroplasticity switches are really turned on. But as you know, because the brain and the oldest part of the brain, or the amygdala, likes to process negative events to keep you safe over positive events, any negative or traumatic event that happens during that brain development period, 0 to 3 and 10 to 14, will massively wire the brain in a negative way and change the structural and functional properties of that part of the brain. So the other period of time is 10 to 14, and we all know about the teenage brain. So that sounds like there's no hope, but that's just not true because of neuroplasticity. Neuroplasticity is the ability to change the brain forever, and the brain has a massive capacity to do that. As neuroscientists, we taught people that the brain is fixed is plastic up until about the age of 25, but then becomes fixed and difficult to change. I think the jury is out now saying that that is just not true. Although it is more difficult to change the brain after the age of 25, the brain definitely has a massive capacity for change. So for these reasons, we have to look at what we have in front of us. We have to first of all understand our own brains so that we can become more empathetic and compassionate to the brains that we're looking after. People are using drugs, taking sugar and over-consuming or gambling for a reason. And my current hypothesis, and where I've moved my research lab, is to understand not just the substances on the addiction side, but also the underlying causes on the stress side. And that is, how do adverse childhood experiences impact the intake of sugar and alcohol down the track? And then, of course, I'm now really moved my lab into neuroplasticity, because I believe that it's going to be a holistic approach where medications can help in certain cases and can be an adjunctive therapy, because I am a pharmacist by training and I spent a long time developing medications, so I have seen their beneficial effects. But what I see also is, and I see that Sovereign Health is doing this, where you have an integration of services. We have to include the principles of neuroplasticity into our practices if we really want to make a change, if we really want to help people, rather than having them come in and out and back and forth. This is why diets don't work in the long term also, because we're not making the fundamental brain changes needed to drive the long-term changes that we want to see both in the brain and the body. So I want to conclude now, and I've left more time for questions if there are any. I just want to tell you that I wasn't always going to be a neuroscientist, except that my sister got schizophrenia. That made me go back in 1990 to learn more about the brain. And I've been developing tools and techniques and looking for better approaches to the treatment of mental health problems and addiction ever since. And I continue to do that. So my goal is to help 1 million people take back their brain power by applying the principles of neuroplasticity and getting to know and understand their brain more than they have in the past. I want to thank you for listening to my presentation, and I hope that you're able to get some interesting information. I'm happy to take any questions, and I hope you enjoyed my presentation. Thank you.
Thank you, Dr. Bartlett. That was a great session, I must say. Thank you for this presentation, for this splendid presentation. Okay, now, just as a reminder for everyone attending this webinar, please use the question answer box provided in your screen, like it's just underneath your presentation, your slide box. You can use that box to send in your questions if you have any questions to ask to our speaker today. Dr. Bartlett, we have a question from Gary. I don't see much written about stevia extract as a sweetener. Is it much safer than sugar?
Oh, yes, I get asked that question a lot. I actually put in, I'm going to try and go back. Yes, so we did the saccharin experiments alongside sugar experiments, and I actually put that slide in there to show you that the same effects we observed with saccharin in addition to sucrose, which we're really surprised about also. Saccharin consumption, and we showed that saccharin could change nicotinic receptor expression. They were questions asked by the reviewer when we submitted the paper. So that was a great question. And honestly, that was really surprising too, because we were using sucrose to match the ethanol and then thinking that it might be just the sweet driving the changes. But then when we saw that saccharin had the same effect, we were really surprised with that observation. So this panel C down here, I don't know if you're still looking at the presentation, but that's actually saccharin consumption there. So you'll see here sucrose consumption on the top panel 4 weeks, and you'll see saccharin down here. Okay, next question.
Okay, the next question is from Biljana. Is there a correlation with stress and salty foods?
Yes, we haven't done that yet, but high fat, definitely we know that high fat containing foods, which often have salt, drive the same brain changes that sugar does. Other people have seen that too. So not everyone has sugar. Some people, so we actually look at high fat and sugar. Today was all about sugar, but I think high fat foods has a lot to play with it as well. I think they go hand in hand.
Okay, the next question is from Kevin Brown. Oftentimes, drug and alcohol counselors will encourage patients to keep a candy bar handy in order to not go hungry or do address periods of low energy. What do you think about these suggestions for those early in recovery?
Yes, Alcoholics Anonymous has done the same thing. Well, now you know why it works. So now you know why people are given, why sugar works to quell the cravings during alcohol withdrawal period. It's substituting, it's giving a nice little boost to endogenous acetylcholine, because when people are coming off alcohol, they've got to go through a hypodopaminergic period where there's dopamine deficit, and they have to rebuild all those proteins that have been absent. So it's no wonder sugar can help. Now we know why. People have always been told to suck on something sweet if they're craving. So my bottom line here is, depending on their diabetes, whether they have diabetes or their sucrose level, I think that if something can help them to overcome going back to alcohol, then I don't see a problem with that, because all of the alcohol is just far worse than being addicted to sugar, in my view, in the beginning stages of coming off alcohol.
Okay, thank you. Next question is from John. Natural sugars in fruit and honey are just as harmful also?
No, they're different. They're metabolized differently. They're not the same as fructose from high fructose corn syrup or processed sugar. They're metabolized in the body. The way they're metabolized in the body is quite different. And I didn't put any of the information about how the energy from fructose is burned by the liver and stored in visceral fat cells around the stomach and things like that. That's why people get a beer gut, or people put on weight around the stomach, because the energy from fructose is actually stored in visceral fat cells, and those cells multiply and change. This is what I wrote in my book also, under print, all about how fructose affects our body as well as our brain.
Thank you. Moving on to the last questions. This is from Dennis. How much sugar for humans, and over what period of time does one get the same results as the animal research?
Yes, people have asked that question before, and it just depends on when it all started, like what age. Just like everything, the earlier you start, the worse it is for your body. It's hard to replicate because animals live for a much shorter period of time, and we don't know exactly the amount of time it takes. But I think, just think of yourself, and you know that if you want to have one piece of chocolate, it'll lead to two pieces of chocolate, lead to a chocolate bar within a certain period of time. So that's just an example of how the brain needs more to get the same level of reward. So, and I just want to say that this is really important, that every person you're dealing with is very different. There are no hard and fast rules here for everybody. So everyone has a different adverse childhood experience score, experience, a different brain. And so what might take one person can have a piece of chocolate for their whole life and be fine. The next person might have a bar of chocolate straight away and then a tub of ice cream, for example, on the sugar side. People on the alcohol side, we know the younger you start drinking, the worse the outcome. So every brain, there's a reason for everything, and each person is so different.
Okay, there is one more question from Michelle. Has it been proven that artificial sweeteners can trigger more of the carb craving? Do these products have a place at all when it comes to those that are addicted to sugar?
Because of the way these things activate the brain, my view, or what I've seen in people, is that you can replace them, but then they lead to the same problem. So the only way forward, just like with alcohol, if someone's addicted to alcohol, they go through a withdrawal period to have abstinence so their brain can reset. People that are really addicted to sugar, not just people eating one piece of chocolate, but people that are really addicted to sugar like alcohol, there's no other way but to come off it so that we can reset the hypothalamus, the prefrontal cortex, and the amygdala slowly. Come off it, reduce it slowly so the body can reset and the brain can reset. This is how strong a chemical it is on the brain.
Thank you. And moving to the last question of this session, from Nancy Pitts. I have been in a 12-step program called Food Addicts Anonymous for 10 years, and I'm maintaining a 95-pound weight loss for 9 years. Do you refer clients to this 12-step group? We do not eat sugar, flour, or quantities.
Yeah, well, that's exactly right, because as I said to you, the sugar, sugar, flour, and some of these other things, depending on what it is, are activating the brain, right? And so they make you hungrier for other foods. And they go into the visceral fat cells, the part of the body that's the hardest to move. So it's a double whammy. I like to say it's something like this. You go to you have to exercise as well, but you know that if you go to the gym and then you reward yourself afterwards with a Frappuccino, the calories from that Frappuccino are going to take you another 4 hours at the gym to get rid of. Only if you went straight back to the gym, because the way that energy is stored in the body goes into these cells that are like cancerous cells that keep developing and multiplying. So the only way to maintain weight in the long term is by reducing sugar intake, managing stress, and doing brain training, brain plasticity, by resetting the brain also. Because that's key. The key is the brain training. Comes the brain first, body second.
Okay. Thank you so much, Bartlett. Now we need to end this session right here. Keeping the time frame in mind, we need to end this session. Whatever questions are unanswered, we'll try to send send all the questions individually in the email boxes.
Oh, good. Okay.
All right. Thank you so much. Thank you so much, everyone, for your participation and joining us today. If you have any further questions, please feel free to send them to webinar@solvehealth.com. I repeat, webinar@solvehealth.com. If you have any questions related to CE certificate, please send your queries at ce@solvehealth.com. I once again repeat, ce@solvehealth.com. Once you leave today's webinar, you will receive a thank you email within 24 to 48 hours with all the information pertaining to this webinar and how you can earn your CE certificate. And lastly, a survey will appear on your screen once I end up this session. So I request you all to fill that survey and give your valuable feedback. Thank you so much for joining us today. Thank you so much for your participation. We hope you found this information valuable. Have a great rest of your week, and we'll look forward to bring more interesting webinars each month for you.